Hi gang, welcome to Anatomy Physiology video 10.05, where we're going to take an overview of the membrane proteins that are associated with nerve impulse transmission. By the way, the other term, what we call when we conduct a nerve impulse, is action potential. So we're working our way up to understanding action potential. But just like we did with sliding filament theory, which is a pretty complex physiology, in order to understand action potential, there's a lot of microanatomy that we have to go over. Now, this video, and actually the next three videos, this one and the next two, are review from biology. You spent quite a bit of time in biology looking at membrane proteins that were embedded in the phospholipid bilayer of the cell membrane. So this hopefully will come back to you as we go. Let's break this down piece by piece, though. And to start with, let's review the phospholipid bilayer. So if you remember, the cell membrane has phospholipids, and they form a bilayer. And that's what we see here. So if you look at this, in my diagram, they're orange with yellow. This is our phospholipid bilayer. And inside this phospholipid bilayer of every cell are proteins. And they stay suspended within the bilayer like this. So they operate as barriers to what can go in and out of the cell. So they control what enters and what exits the cell very often. So they're a gate in a sense. Well, before we can talk about how substances get across, we have to review the forces that cause substances to cross. And I want you to draw a nice stark line here in between this column and these columns. These represent two categories of membrane proteins. On this side, everything on this side, substances crossed by active transport. Active transportation. Active transportation. Everything on this side operates on passive transportation. Or passive transport. Pardon me, I'm getting over a cold and this cough just can't quite shake it. So what does that mean? What's the difference between passive transportation and active transportation? Well, it has to do with concentration. So let's review concentration and concentration gradient. So I'm going to draw two circles. And I'm going to put a substance or a solute in one. If I connect these with a tube, so let's say it's a gas. Let's say I made two flasks and this has argon gas in it, and these are all molecules of argon, and these are molecules of argon. When I open up the stoppers between these two, what's going to happen? Well, I hope that you said we'll get flow of our gas in this direction. Because if we had to talk about this in terms of concentration gradient, we would say that this has got a high concentration and we would say this has a low concentration. Concentration gradient just means that we have different concentrations in different areas. Different concentrations in different areas. I'm going to abbreviate to save space. That's all concentration gradient is. Now, after I let this flask set for a while, what we would expect it to look like So it's still open. And hopefully you can see here, we really don't have a difference in our concentrations. So now we have the same concentration in both. And if I get one that flows this way, another one's going to flow this way. So we have equilibrium. Well, this is exactly what happens in cells. And if this represents the cell membrane, so here, this is the cell membrane, and usually it's blocked. You can't cross. If I have a difference in concentration outside the cell, and by the way, here in my diagram, this is outside the cell, this is inside the cell. This is a before, and this is after. So this is outside the cell, and this is inside the cell. So here my membranes are closed, here it's open, closed, open, closed, open. So before my membrane, and this would really be better served, let's do 6, 1, 2, 3, 4, 5, 6. So I've got 6 of these ions outside the cell, and my membrane's closed. Just like this, I have a higher concentration. Then the membrane opens, and it can begin to flow, and when I'm done, I'll have 3 inside and I'll have 3 outside. So I get an equal amount. So I go from having a concentration gradient to having an equilibrium once this protein gets opened. Now, how that protein gets opened is the next lesson. We call this let's define passive transportation. It's a movement of a substance from high concentration, and let's add automatic movement of a substance from high concentration to low concentration. Or we call this moving with the concentration gradient. Moving with the concentration gradient. Why does this happen? If you've ever been outside a store on Black Friday and they didn't have crowd control, and the people were all packed up in front of the doorway, then when the doorway opens, people in the back are aware that it opens and they begin to push, and there's a pressure between the people, and it actually pushes people through the door. If you're not careful and you trip, there have been stories about people that get hurt on Black Friday. Well, molecules and people really aren't all that different. We have all these molecules around, or ions around the outside of the cell running into each other, pushing against each other, whereas here there's no pressure, there's no push. So when this door opens, when this gate opens, it's the force of these molecules hitting each other that pushes them through. And now when we have equal amounts on both sides, we have equal forces. And each of these 4 proteins allows substances to cross this way. Now, they all have a different way of opening, but whenever we have substances on this side and this side, this is how they get through. Over here, though, we have something a little bit different going on with active transportation. Let's look at our particles. So this is before the protein opens. This is after, and it's embedded in a phospholipid bilayer. And to start with, I've got 2 particles outside the cell, and I've got 2 particles inside the cell. This is out and this is in. But down here, look what happened. I went from having 2 particles outside the cell to having all of my particles outside the cell. So if I went back to my original example, if I have my 2, let's do blue. Now I've got my 2 flasks, and I've got a high concentration. I've got a low concentration, and I'm going to connect those. This is what happens with active transport. So it would be as if I open up the tube, and when I'm done, so this is before, and this is after. I didn't get a distribution of my particles. In fact, I didn't get less concentrated, I got more concentrated. Things became more concentrated. So if this is high concentration and this was low, I actually went the opposite direction. My flow went from low to high. So I went against the concentration gradient. And folks, in nature, this would never happen unless I put in energy. So this only happens as the result of energy input. And the energy that we're always talking about in biology and anatomy is ATP. So it requires ATP. Because think about it, these guys are knocking each other around and they want to get away. And I'm actually increasing the force that exists between these particles. So it actually required me to overcome that force. And to do that, I had to have an energy source. So active transportation goes against the concentration gradient. But it requires the input of energy, and we move substances. We move substances from a low concentration to a high concentration. I'm going to abbreviate concentration. So what are our takeaways? When we're talking about membrane transport, right off the bat, you have to understand, are we talking about passive transport or active transport? Passive transport, we're moving things from a higher concentration to a lower concentration, and it just happens, key word here is automatically. Automatically. Because nature wants things evened out. It wants an equal amount of force between the particles on the outside and the inside. If we're talking about active transportation, we're taking a higher concentration and a lower, and we're actually making it more concentrated. That's called moving against the concentration gradient, and this has to have ATP. So the two forms that we're going to talk about, the next video, we're going to talk about passive transport. We're going to do these ion channels. And we're going to talk about how do these ion channel gates or doors get opened? Because we have to open them. There's different ways you can open them. And then in 1007, we're going to talk about the mechanics of, this is actually the sodium potassium pump. Sodium potassium pump. And so that's 1007. All right, so let's go on and talk about ion channels and how we'll get them open.